Hello, everyone, and welcome. I'll just give folks a few minutes to get logged in. Hope everyone's having a great day. Okay, so I think I will get us started. So good afternoon, everyone, and welcome. My name is Caitlin Jaggers, and I am the research lead for the Research Knowledge Translation and Exchange team at the Alzheimer Society of Canada. The Alzheimer Society of Canada is very pleased to welcome you to another ASRP Exchange presentation that is being offered through our provincial support program. This series has been developed to feature ASRP recipients who have completed their funding and who will speak to the outcomes of their ASRP supported research while addressing their results and how they will continue to impact people with lived experience. Joining us today is Sarah Sarovich, the Research Program Assistant at the Alzheimer Society of Canada, who will provide a brief introduction for our presenter and walk us through today's agenda. We also have Shirley Lucas joining us from the Alzheimer Society of Newfoundland and Labrador, and Shirley will be responsible for moderating today's question and answer period following today's presentation.
Thank you, Caitlin. Hello, everyone, and welcome. As Caitlin mentioned, my name is Sarah Sarovich, and I am the Research Program Assistant at the Alzheimer Society of Canada. I would like to start off today's presentation by going through the agenda presented here, and then I will pass it off to our presenter, Dr. Matthew Parsons. Before we begin, please note this webinar is estimated to be under 1 hour in length and is being recorded, but that only the video and audio of the presenters is captured during the recording. The presentation recording, in addition to the PowerPoint slides in PDF form, will be made available on Connection for Alzheimer Society staff, as well as on the Alzheimer Society of Canada website on our ASRP Exchange webpage following today's webinar. Today's webinar will be presented by Dr. Matthew Parsons and is being offered as one of the ASRP Exchange webinars within our provincial support program, Meet the Researcher series. We are so pleased to have Dr. Parsons here with us today to present his project, Strengthening Brain Cells, Determining the Failure of Brain Cell Communicating in Alzheimer's Disease. This project was funded by the Alzheimer Society Research Program, the ASRP, starting in September 2017, with project completion ending in August 2021. Dr. Matthew Parsons received his PhD from Memorial University in 2011. From there, he moved to Vancouver, where he worked as a postdoctoral fellow in the lab of Dr. Lynn Raymond at UBC. While at UBC, Dr. Parsons studied the cellular side of Huntington's disease, a devastating neurodegenerative disease characterized by a triad of motor, cognitive, and psychiatric symptoms. Dr. Parsons is primarily interested in identifying what goes wrong with brain cells before they die. In most neurodegenerative diseases, brain cells lose their ability to communicate with each other before they die. Therefore, Dr. Parsons seeks to understand the nature of this communication breakdown in brain disease and hopes to identify ways in which we can prevent cell death by restoring healthy communication. Dr. Parsons started his own lab back at Memorial as an assistant professor in 2015, and in 2020, he was promoted to associate professor. Work from his lab appears in highly regarded peer-reviewed neuroscience journals, and he is the recipient of notable research awards from Memorial University, including the President's Award for Outstanding Research, the Dean of Medicine's Excellence Award, and the Suncor Terra Nova Young Innovator Award. His research is funded through a variety of sources such as the Natural Sciences and Engineering Research Council of Canada and the Canadian Institutes of Health Research. Notably, a research grant from the Alzheimer Society of Canada allowed Dr. Parsons' lab to investigate brain cell communication failure in the context of Alzheimer's disease. Their findings related to this project will be the focus of today's webinar. As mentioned, at the end of today's presentation, Shirley Lucas will be moderating a short question and answer period. Please use the Zoom chat box feature if you have any comments you would like to make or questions you would like to pose during the presentation. Please note we will hold off on asking our presenter to respond until after he has completed his presentation. And without further ado, please join me in welcoming Dr. Matthew Parsons.
Okay, thank you very much for the introduction, Sarah. Thank you. Okay, so I will share my screen and we'll get right into it. I can still see some videos. Can somebody just confirm that you can see my full screen of the title page and then this laser pointer going around? All right, thank you, Sarah. Okay, so this is my talk on what we've been doing in the lab with regards to this project over the last couple of years. I am, and my lab is a cellular neuroscience. I'm a cellular neuroscientist. My lab is a cellular neuroscience lab. I assume all of you are not cellular neuroscientists. So I have the challenge of kind of summarizing our research findings, knowing that you don't have decades of background in cellular neuroscience, which is what I do. So I'm going to take this approach to really kind of get us all on the same page to understand what a synapse is and how brain cells communicate, and most importantly, how they fail to communicate in the context of Alzheimer's disease. So first of all, just a huge thanks to the Alzheimer Society of Canada. Thank you to Sarah for the wonderful introduction and to Caitlin for putting this together. And of course, the ASRP and all the donors. Without you, none of this work would be possible. So I guess Alzheimer's doesn't really need much of an introduction to this group. It's the world's most common neurodegenerative disease. And I highlight that word because I don't particularly like the term. It really puts the focus on the fact that cells die in the brain. And that's true, they do die in the brain, but a lot happens before they die. And I don't like the focus on the cell death because a lot of communication breakdown occurs before cell death. And I think that really should be the focus of the disease, because replacing brain cells is a really difficult task. So I have this image here taken from a 2015 publication, and I just ask, what is this? You don't have to put on your microphones and say anything, just think about what this could be. The answer is that it's a single brain cell. So the cell body would be down here, if you can see my laser pointer, and then it sends a single axon out. That axon then branches off, and then the branches then branch off. And by the time it gets to its target area, it's actually making tens of thousands, perhaps hundreds of thousands of contacts just from this one single brain cell. So you can imagine if you were to lose that cell, replacing that cell as well as all of its contacts would be a very difficult, if not impossible, task. So the key here is that before these brain cells die in Alzheimer's disease, they lose their ability to communicate with each other. So I want to think about, and I want you to think about Alzheimer's disease as a brain cell communication disease, a synaptic disease, rather than a neurodegenerative disease. That shifts the focus earlier in the disease. While we still have the cells that are there, the communication is just what's starting to break down. And I think that's the key point there that we need to learn more about. So in order to get us all on the same page, we all need to know what a synapse is, and we all need to know the basics of how a synapse works. So how do brain cells communicate? Well, here's a nice cartoon image of the human brain. And it's really hard to understand the brain's complexity just by looking at a brain with the naked eye. So again, you don't need to turn on your microphones and say anything, but just take a guess at how many cells and how many connections are in your brain. So think about your brain. How many neurons are in your brain and how many connections do they form? So it's impossible to get an actual answer for this, but estimates are in the ballpark of 100 billion neurons and 100 trillion connections. So those 100 billion neurons make about 100 trillion connections. These are unfathomably high numbers. And it just really shows how complex the brain is. And this is what we're dealing with. We're dealing with an organ that has 100 trillion connections, and each of those connections is referred to as a synapse. And that's what we're concerned with, is that what happens to those connections? What happens to those synapses before the cells die? So in order to understand the complexity of the brain, we can't look at it with the naked eye. We have to go under the microscope. So in my lab, we do things like we take brain tissue. This is an area known as the hippocampus, and we put it on microelectrode arrays. These are just simply electrodes that monitor neural activity within the tissue. We can take it and look at various proteins. We can stain with using red and green labels and make pretty pictures and get an idea of how much protein is expressed in certain areas. And you can see down here, this scale bar, that scale bar is 250 microns. So that's 1/4 of a millimeter. So we're zoomed in quite a bit, but nowhere near far enough to see an individual synapse. So to understand synapses, we have to get in even closer. So what we also do is some morphological staining with this Golgi stain, and this is a dendrite. So we'll get into dendrites, we'll get into pre and post synapse soon, but what this is kind of the receiving end of a connection. This is a dendrite, and you can see the dendrite is studded with all of these little things that we call spines. And those spines are kind of the receiving end of a synapse. So they receive a signal. So each of one of these spines represents the receiving end of a single synapse. So you can see even in this really short dendritic segment, we have tons of these spines happening, and contacts are happening on all of these spines. So just the basics of communication, let's just break it down to as simple as we possibly can with two cells. Cell A wants to communicate with cell B. So how does that happen? So cell A will send an axon to cell B, where it will synapse with one of these spines, one of those little studs that we saw on the last on the last slide. And if we zoom into that, that is a single synapse, and we have 100 trillion of these. And these connections completely define who you are as an individual. Your memories are stored there. Your personality is encoded by these synapses. Without them, neurons are useless. If you take away this connection and cells exist in isolation, they may as well be dead. Without the connections, without the synapses, neurons might as well be dead. They absolutely need to communicate to work. And this is what happens in Alzheimer's disease. You start to lose these connections, and that happens before the cells die. So we need to figure out why we lose these connections and what we can do to prevent it. So this is as close in as we can get. This is an individual synapse. And I'm going to come back to this image after we're all synapse experts, so you can identify what is what in this image. This is an electron microscope image of a single synapse. And we have 100 trillion of these in the brain. So before we come back to that picture, I just want to go over the basic requirements for a synapse. And I like to use this example from the Close Talker episode of Seinfeld, where Elaine's boyfriend gets uncomfortably close to somebody whenever he's talking to them. So in this case, what we need for communication, we need a speaker, we need words, and we need a listener. And a synapse is not that different from this. It can be as simplified, it can be as simple as this, where we have a presynapse, which is the speaker. We have some sort of chemical. The most abundant chemical that's released by neurons is a chemical called glutamate, and that's the one that I'm focusing on here today. And then we have a postsynapse, and that's the listener. So those are the three things that you need to know to be synapse experts. That we need a presynapse, which is a speaker. We need glutamate released by the presynapse. Those are the words. And we need the listener, which is the postsynapse. So now if we go back to this image, now that we're all synapse experts, where do you think the listener is and where do you think the speaker is? Again, you don't need to turn on your microphones. Just think about it for a second. What's going on? There's a lot going on in this image, and it might be hard to figure out. But the speaker in this case is this part right here. And the listener is this part right here. So how do I know that? The main giveaway is the fact that the speaker contains all these little packages, these pouches of glutamate. It's glutamate that is in these little packages. So this is the presynapse, this is the speaker, and it's going to release the contents of these packages into this little tiny area called the synaptic cleft or the synaptic space. And it's going to initiate some response in this cell here. This is the listener. And this dark band just goes to show that the listener has a lot of different proteins here that are ready to respond to that release of glutamate. So if you can look at this and say, how can we possibly have 100 trillion of these in our brain? Well, it's because neurons are very close talkers. So to put this in context, the width of the synaptic cleft is 20 nanometers. So this space between the listener, the speaker and the listener, the presynapse and the postsynapse, that space is 20 nanometers. Now in comparison, the width of a single human hair is 75,000 nanometers. So we're talking about minuscule distances here between the pre and the post synapse. So we're really, really zoomed in here. But this is the level of detail that we need to understand what goes wrong in Alzheimer's disease before the cells actually die. So this isn't meant to scare anybody. This is meant to show that even when you look at a single synapse, it's incredibly complicated. These are all the different forms of proteins and enzymes that are known to exist at the presynapse, at the speaker, and at the postsynapse, at the listener. And there are 100 trillion of these in the brain. So it kind of begs the question of why is it so complicated? Why are all these proteins required? Well, we need all these proteins because synapses need to be flexible, because as humans, we need our behavior to be flexible. As a very simple example, think about if you moved. You just bought a new house, you moved. You go to work, your first day at work after the move, you leave work, you have to go home. What home do you go to? Now you've been driving to house A for years and years, but now you live in house B. You need to rearrange those synapses. You need that behavioral flexibility to say, oh no, I need to turn right at this stop sign instead of left. Left is the old house, right is the new house. You need your brain to be flexible to adapt to changes in your situation. So the brain is extremely complex. Even when you look at an individual synapse is extremely complex. And this is really the level, like I said, of detail that we're looking at when we're looking for what goes wrong in the context of Alzheimer's disease. So the big question here is, why do we care about the synapse? Why am I here talking to you about these individual connections? Well, it is very well established now that in Alzheimer's disease, a synapse loss is the best correlate of cognitive decline. So here we have a paper from 2018 where they actually found a great way using PET imaging of this fancy sounding protein called synaptic vesicle glycoprotein 2A. All it is, this is a protein that's found at synapses. And they found a way to tag it and look at it in a PET scanner. And what you can see is that here in the healthy brain, you have plenty of hot colors, the hotter colors, the reds, the orange, the yellows. That means more synapses. If you compare the healthy brain to an individual with Alzheimer's disease at the same age, you see a lot less of the hot colors. You see a lot less red here, there's more green. So it's a lower density of synapses. So we do now have this kind of measure of synapse density that we can do as kind of a marker of synapse loss. And looking at synapse loss, this is again from the same paper, is that there's a great correlation between episodic memory score and this measure here, which is basically translates to synapse density. So the higher the synapse density, the more synapses you have, the better performance you'll have on these tests of episodic memory. So more than anything else, more than amyloid burden, more than tau burden, synapse loss represents the best correlate of cognitive decline. The more synapses you lose, the faster and the worse your cognitive decline is going to be. So the million-dollar question is, how are synapses lost in AD, and how can we save them? So let's just get back to, how does the synapse work? So we have presynapse, postsynapse, the speaker and the listener. So I need you to remember those two things. And then we also have the words. That's glutamate. That's the big thing that you need to remember, this chemical that's released by the speaker. So what happens when the presynapse, when the speaker speaks, what actually happens is that these packages of glutamate, they're contained within these little tiny membranes. It's called vesicles, synaptic vesicles. They actually fuse with this membrane here to the point that it's the transmitter, the glutamate, actually spills out. And in addition to the transmitters, we have these receptors. The receptors come in different shapes and sizes. There are different types of them. They're located both on the speaker and on the listener. And it's the receptors that actually see or sense the glutamate, and then they respond. They might let in some sort of current, or they might initiate some sort of intracellular change here. The bottom line is that it is a chemical transmitter that initiates a change in the behavior of the listener, of the post synapse. So this is the main way through which neurons communicate. The thing is that it's a very delicate balance. Achieving proper synaptic communication is a very delicate balance, and we have to hit that kind of Goldilocks area of just the right amount of glutamate release. When you do have it just right, you get rapid cell-to-cell communication. So glutamate's absolutely essential for life. You absolutely need it for almost everything your brain does. You need it for rapid cell-cell communication, for fast processing, for network stability and flexibility, for strong cognition. You absolutely need the right amount of glutamate released at your synapses. Unfortunately, if you have too much of a good thing can be detrimental sometimes, and that's certainly the case with glutamate. Too much glutamate can lead to toxicity. It can actually kill cells, and without the neurons, your cognition will absolutely suffer. On the other hand, too little is also not a good thing. Too little glutamate release, you're going to have poor information transfer from the speaker to the listener. You're going to have slow processing overall. And the brain can be a bit unfair sometimes, we'll say. It has this kind of use it or lose it attitude, where synapses that are weak, the brain has mechanisms to actually just get rid of it. So the brain looks at all the synapses and says, okay, these synapses over here, they haven't been active for days. Let's just get rid of them. So this use it or lose it attitude is a way for the brain to get rid of unused or very weak synapses. And I highlight this because I think this is really what's happening too much in the Alzheimer's disease brain. And of course, you lose your synapses, it'll result in poor cognition. So if we go back to the schematic here, what happens to glutamate after it's released? So there's one more thing that you need to know, and then we're all synapse experts. This one other thing is what happens to glutamate after it's released. Okay, so we've had these packets of glutamate that fuses with the membrane. It releases glutamate, binds to the receptors. The postsynapse has a response. So the listener has listened to the speaker, but now these receptors, they don't eat the glutamate. The glutamate doesn't disappear. It has to go somewhere. So what happens is that there's a special type of cell called astrocytes. And we can think of these as sponges that soak up the glutamate. It kind of gets lost in that analogy of the speaker listener, but you can also think of these as like maybe acoustic treatment in a recital hall, something to tame the echoes and to really clarify the actual words. So these sponges are responsible for soaking up that glutamate after it's done its job. So glutamate's released, it'll act on receptors, and then it's soaked up by the astrocytes. So what do we think is happening to these synapses in Alzheimer's disease? So in our healthy synapse, now that we're all synaptic experts, we all know that glutamate's released, it combined with receptors, and then it's taken up, it's soaked up by our sponges, our astrocytes here. That's healthy. That's we've found our just right amount of glutamate for healthy synaptic neurotransmission or communication. What we think is happening in Alzheimer's disease is that the sponges fail. We think that there's an issue with these sponges. And what that leads to is not too much release or too little release. It's just that it's not cleared properly. And we think the major consequence of that is to activate these receptors here that are located on the speaker. Now, I haven't talked about these receptors, but these receptors are there to serve a very important purpose of negative feedback. So what this will do, when these receptors are active, that's a way of telling this speaker that you're speaking too loudly, when in fact it's not. It's not releasing too much. It's just because these sponges aren't working right. We think that there's too much activation of these receptors, which tells the presynapse or the speaker that you're speaking too loudly, and then it shuts off neurotransmission. And then if you shut off neurotransmission, it becomes a weak synapse. And remember, the brain being unfair sometimes, what does it do to weak synapses? It gets rid of them. So we think that this is a major contributing factor to the synapse loss in Alzheimer's disease. So what we think is happening, we think in the AD brain that the sponges fail. That leads to overactivation of receptors on the presynapse, the speaker over here. That makes the speaker think that it's speaking too loudly, even though it's not. So it responds by lowering its voice, meaning it releases less glutamate. Then it becomes a weak synapse that, if it's sustained, it's eventually eliminated. Okay, so this is all great. I can show you these cartoon images of what I think is going on. But what evidence do we actually have that makes us think that this is what's happening? So the next few slides, I'll show you some of the data of how we got to this point. And if it gets a little technical, I'll make sure that I highlight that main take-home point. But this is kind of the experiments that we've done in the lab to figure this out. So what we do in the lab, one of our major techniques is visualizing glutamate, the words, in real time. And this is a really, really cool neuroscience tool that was developed less than a decade ago, so it's quite new. It's actually pronounced iGluSniffer, and it stands for Intensity-Based Glutamate Sensing Fluorescent Reporter. And really what you need to know is that it's this green protein, and it lights up when it senses glutamate. So think about it as, you know, one of those motion detector floodlights that you might have on your house. When it sees motion, you know, somebody coming onto your property, the lights will come on. Basically, if glutamate comes into the area of this, it will light up. So it's kind of like a motion detector, but for glutamate, and it's green. So this is what it is. It's actually made up of two components. It has this part here, which is the glutamate binding site, and it's fused to this part here, which is basically the green light. So when it sees glutamate, it makes this kind of change in the shape of it, and that just switches on the light. So we can look at how much green comes through our microscope, and we can translate that to mean the levels of glutamate acting in that vicinity, in that area. So we can introduce this GluSniffer into brain cells, and by doing so, we can tell how much glutamate is released and how long it stays around. So how well those sponges are working to get rid of glutamate once activity has ceased. So I put a little asterisk there to make us think, okay, how am I getting a green motion detector of glutamate into a human brain? Well, that's the thing, that I'm not. And this is something that is, maybe we can have a bit more of a discussion about this later on in the discussion section. But this is a caveat in a sense, is that all of the work at the synaptic level, looking at really detailed mechanisms, it's very difficult, if not impossible, to do that in humans. So this is the importance of animal models of Alzheimer's disease. And I've worked with a lot of Huntington's disease animal models, and now with Alzheimer's disease animal models. There are a whole bunch of different ones. They all have their advantages and disadvantages, but always keep in mind, it's very difficult. Even just the nature of this is that human Alzheimer's disease, usually you don't see any symptoms until multiple decades have passed. So it's very difficult to model that progression in a mouse that has a lifespan of about 2 years. So that's a huge disadvantage there right off the bat, is that the lifespan actually doesn't quite match up at all. But these models do a very good job of mimicking a lot of the pathology associated with Alzheimer's disease. And once you get down to the level of an individual synapse, mice are certainly not humans, but the components of a synapse are very similar, strikingly similar between mice and humans. So what I mentioned about the speaker and the listener and the sponges and the glutamate and the receptors, all of that is very similar, if not identical, between the mouse and humans. So we can learn a lot about synaptic function, and we can learn a lot about synaptic dysfunction and disease from animal models. So this one that we use is called the 3XTG mouse model of Alzheimer's disease. They have these mutations that are linked to familial forms of Alzheimer's disease. And as you may know, these familial forms, the inherited forms of Alzheimer's disease, certainly make up only a very small minority of all cases. But those mutations do drive amyloid and tau pathology. I'm sure these are words familiar to most, if not all of you. And importantly, these animals exhibit synapse weakening over time and eventual synapse elimination. And we know that this happens in human AD. And they also exhibit cognitive deficits. So not right away, not when they're first born, but kind of midlife, if you run them through certain mazes, and that they won't perform nearly as well as control animals that do not have these mutations. So there's a stunning amount of research done on this model and other types of models. We know so much about little tiny details in these models, and we're learning so much about Alzheimer's disease in general because of these mice. But always that caveat is there, is that because it's true in the mouse doesn't necessarily mean it's true in human AD. It means that it's worth continuing looking into. So this one is one of the most commonly used ones. And I put an asterisk here just to say different models could be discussed further in the Q&A session to follow. I find it a very interesting area, and because right now it's really the only way to get such detailed understanding of the synapse and how it fails when you have mutations that are known to cause human forms of Alzheimer's disease. So if we go back to our synapse, now we're talking about a mouse. This is a mouse synapse, looks a lot like our human synapse. So what we can do now is we can introduce this detector, that glue sniffer, into the speaker. What we've done is introduce it specifically in the speaker, not in the listener. We're interested in this area here. So we've introduced a detector into the presynapse. So any glutamate that would normally activate these receptors, and remember, these are the ones that tell the speaker that you're speaking too loudly. Any glutamate that acts in that vicinity, it's also going to trip our detectors. So anytime we see green light, we know that enough glutamate is in this vicinity to hit those receptors to cause that negative feedback regulation to shut off neurotransmission. So these are just a couple of data slides, and I'll reiterate the take home after I go through them. But this is a very early disease stage in the AD mice, 3XTG, triple transgenic, we call them. But these are this is the Alzheimer's disease mouse model. So what we do, we take brain tissue from these animals that have that motion, that green motion detector. Then we stimulate neural activity. We use an electrode to stimulate neural activity. And then we look at levels of light, the level of green light that the tissue gives off. And then we can measure it. And then we can see how high it goes, so how much glutamate is released when we stimulate. And then when we end the stimulation, how fast does it take the brain to clear that? How long does it take the sponges to clear that? And this is at a very early disease stage. These mice have all of the brain cells that the control mice have. There's no cell loss. There might be subtle, very subtle behavioral deficits if you were to look on certain very sensitive tests. But just to look at them, absolutely, they look just like normal mice. So if we do this, you can see that the control animals, the traces in blue, that green light goes back down to baseline, so it turns off much faster than it does in the AD animals. And if we quantify this and repeat it over and over, we see that not quite a twofold, but a 1.5, 1.75 fold increase, almost a doubling the time it takes to clear glutamate in the AD brains compared to the controls. So this would be the clearance rate. How fast are those sponges removing glutamate? Fast is good. Remember, we want a quick glutamate signal, then we want to get rid of it. Slow is bad. Too much glutamate hanging around, not a good thing. So even though the release is the same, you can see that these peaks are about the same. So there's no trouble release. They don't release too much. They don't release too little. At this early disease stage, it's just hanging around longer. Now this trace here is about 2 seconds in length, so we're talking really short time. So this is a millisecond, a couple of milliseconds, tens of milliseconds in the difference. But that makes a huge impact in the end. If we have chronic overactivation of these receptors, because remember, our detectors are right next to them, we think that these receptors are becoming just slightly overactive, but for a long period of time. And that slowly over time just shuts down neurotransmission. So if we take these same mice and look at a later disease stage, this is what the responses look like. So again, we have the AD mice in that kind of yellow goldish trace, and then the controls are in blue. Again, you can see that the clearance is a bit slower, so the sponges are taking longer to get rid of it. But in this case here, you can see that not a lot of glutamate is released, nowhere near what we see in the healthy control animals. So we think that this prolonged activation of these presynaptic receptors are starting this negative feedback cycle that results in this kind of reduction of glutamate release later on. So we can quantify that as the peak or the amount of glutamate release, and it's reduced to about 60% of what it used to be before. So we need better clearance. We need clearance. So what we used is this drug called ceftriaxone. So this is an FDA-approved antibiotic, and it's known to increase the sponges in the brain. More specifically, it's known to increase a protein in those astrocytes that is very heavily involved in removing glutamate. But for our purposes, let's just think of it as a drug that increases the sponge in the brain. So we repeated these experiments in control and AD mice treated with saline. And again, we found that the clearance rate was slowed at that presynaptic membrane at the speaker in the AD mice. But in AD mice that were treated with ceftriaxone to increase those sponges, they were slightly actually faster. It wasn't significantly different, but a little bit even faster than controls. They were certainly much better than the AD mice that didn't have ceftriaxone. So we think this is a good way in the lab, and we'll get to what this could potentially mean in human AD. But in this case, we think in the lab, this is a good tool to speed up glutamate clearance. And right now, we're interested in figuring out better ways or more selective ways to increase the efficiency of those sponges to get glutamate back to control levels. Something that's a bit more selective than ceftriaxone, because this drug is fairly dirty in that it does a lot of different things, including one of the things that it does is has this beneficial effect. So we'd like to follow up on this to see if this now, does this prevent the synapse elimination that happens later? Does this prevent the cognitive decline? Does it help with amyloid and tau pathology. We can do all of this stuff in the animal models, which is really neat. So to reiterate this, we think that in early AD, the sponges fail. And what that does is it leads to an overactivation of these receptors on the speaker. So we showed this by those longer, the motion detector was on longer, I guess we could say, in the AD mice. That makes the speaker think it's speaking too loudly and then responds by lowering its voice. Because then if we look later on in the disease, the AD mice cannot release enough glutamate. And then we think that that becomes a weak synapse that if sustained, it's eventually eliminated. And we do know that these animals have a lower synaptic density at that later age. So the big thing here is the sponges fail early. So we need to figure out why they do. Why do they fail early, and what can we do to prevent it? So to me, and again, I would be dramatically simplifying Alzheimer's disease if I said that this is entirely what causes it. I think this absolutely contributes to it. And I think there can be benefit from preventing that failure. But I think that there are a lot of other things going on that could also contribute. So it's kind of a bit of a storm, but I think this is one of the main contributors to that early synaptic dysfunction or communication breakdown in AD. So just two more slides and I'll clue up. So am I telling you that ceftriaxone is the cure for Alzheimer's disease? Unfortunately, likely not. I said it's a great tool in the lab, but high concentrations are required, and you would need to take it chronically, a really high concentration antibiotic. And like I said, it affects more than just the sponge, so there's likely to be side effects over time, and certainly at those really high concentrations that are required to affect the sponge. But what it does provide us with a much better mechanistic understanding of Alzheimer's, and that opens up new research paths to try to develop better and more specific treatments. So we're trying now ways to overexpress specifically the protein. This protein is called GLT1, but specifically that protein that's responsible for taking the glutamate into astrocytes. And we're looking at ways to selectively increase that protein in astrocytes. So much more specific than just what ceftriaxone is doing. So we want to know, how do the sponges fail? What is it? Is it amyloid that's doing it? Is it tau that's doing it? What in the AD brain is actually making these sponges fail? And what can we do to protect them? And another question of interest is, what are the exact receptor pools? There are so many different receptor subtypes. They can have different blockers or different drugs that act on these different receptors. So what are the exact ones that are overactivated, and how can we prevent their overactivation? So as a summary, I hope I kind of got across the point that AD is a disease of the synapse. Yes, it's a neurodegenerative disease, but let's not think about it as a disease where brain cells die. Let's think about it as a disease where synapses are lost. And as I said, a neuron without connections might as well be dead anyways. So the synapse loss happens a lot earlier. So that is more important at trying to understand and trying to reverse than trying to regrow cells or introduce new cells and try to make them find their original paths and their original connections. So preventing this weakening and eventual elimination will likely prevent cognitive decline. And I strongly believe that therapeutic strategies that maintain strong synapses are likely to be beneficial in AD and also other dementias, because a lot of what I talked about could apply to other dementias as well. So what is needed, in my mind, I think three things are very important to follow up on this. We need earlier detection. This is a big problem in Alzheimer's disease, is the inability to perfectly predict. We're getting better, but the inability to perfectly predict who is going to get AD. So if this synapse loss happens early, we need early detection. If therapeutics are going to have a better effect, earlier detection is a huge area of research that deserves more attention. We need a more detailed understanding of the synaptic failure. The more detail we have there, the more specific our drugs can be. And then lastly, like I said, a lot of this detailed understanding has to come from reduction models like cell cultures or mouse models. And we need better bridges between mouse models and humans, because what works in the mouse may not work in the humans. We need better models to increase the translational validity of what we find in the lab. So with that, we will be continuing to work on this. And I really, really want to stress how much this grant and this funding has meant to me and my lab. And we will absolutely be continuing to work on this. I just submitted a CIHR application based on this work and how we want to go forward with it. So thank you to everybody who listened here today, and a huge thanks to the Alzheimer Society of Canada. And I'd be more than happy to take any of your questions. Thank you.
Thank you so much, Dr. Parsons. That was a fantastic presentation. I think you did an excellent job breaking down all of the material in a digestible way. And I think you really drilled home some very important points that we can all take home with us. So as mentioned, this is the time to pose any questions or comments that you might have for Dr. Parsons. And you can do so in the question and answer box or the chat box. There are two availabilities for you. And we will just proceed as those trickle in to pose those questions to Dr. Parsons. I think while we wait just for maybe some people to collect their thoughts and think of some things they'd like to ask, I think one thing that I'd maybe like to start with, Dr. Parsons, is you had flagged on one of your slides that there are potentially different models to be used besides the mouse model that you demonstrated. And perhaps you could share a little bit about what those other models could look like.
Yeah, so it's something that is extremely important to understanding of any brain disease. Like I said, just because it's so hard to do all that detailed mechanistic work in humans. So my postdoctoral training, and we still do quite a bit of work with Huntington's disease as well. That one's relatively easy in comparison. So Huntington's disease is a single known genetic cause. If you have an expanded CAG repeat in a certain area, you will get Huntington's disease. So in that case, the animals can pretty well mimic the genetics of the disease. You introduce that gene into that animal and it will get Huntington's-like phenotype. With Alzheimer's, it's much more complicated because, as I kind of alluded to, the familial forms, the ones with mutations that are passed down, I mean, estimates are that they only account for about 1% of all Alzheimer's cases, and then the rest have various causes that may be, gene and environment interactions. So most of certainly the genetic animal models of Alzheimer's disease, they introduce these mutations that are seen in those familial forms. So that begs the question, okay, are we only studying familial Alzheimer's in the lab, or are these research findings generalizable to the entire Alzheimer's population? And it's very difficult to answer that. Even within the genetic models, there are a bunch of different options, ones that have only amyloid pathology, ones that only have tau pathology, and it's very difficult. And one thing as a researcher, when you submit grants, it's to the point where a reviewer of your grant, if he or she just doesn't like that one model or prefers a different one, then it could be enough to trash your grant. And there's a lot of strong opinions on the different models and which is the best for which situation. The bottom line is that there's not a best one. They're not perfect, and they have to be taken for what they are. They are just models. The point is to identify things that could be going on in human AD, and then to validate them in other systems before committing to a clinical trial, which is extremely expensive and extremely time consuming.
Great, thank you for providing that context and that additional information. I will also, I can see that there's not a lot coming through in the box right now, which is okay. I think partly because you just did such a phenomenal job explaining everything to everyone. So I guess maybe one thing on my mind is, I know you said your research is going to continue. Maybe you can just explain some of your next steps moving forward.
Yeah, so a lot of the next steps would be to, so with this, a lot of the novelty comes from the technique, the imaging technique. So what we would want to do is to use this in multiple animal models of Alzheimer's disease. It would be, I think it would be a mistake to go, put in more years into this project into one animal model and not validate it in other models. So we'd like to do a couple of different models, but then again, you're getting into, which ones do you choose? And we have a couple of options there. And I think, if we see the same thing there, what we want to do is go in and look at various ways of increasing the capacity of those sponges to get rid of glutamate. I will say that that's something that, so this protein in the sponges that takes up glutamate, we do know that in human postmortem tissue, the expression of that protein is down quite a bit. So we do know that the protein that's responsible for clearing a lot of glutamate is decreased in human AD. It's just that we can't do that type of imaging in humans. So what we want to do is figure out better ways to increase that protein in those support astrocyte cells that are kind of on the side of the synapse that help clear the glutamate. And we want to be able to speed up glutamate clearance and then see if we do that early on, is that enough to completely basically prevent Alzheimer's in these animals. And then it would have to develop from there if that's the case, and it would develop there to test different models and then try to develop some drug that could be useful in a clinical setting.
That's great, thank you. Definitely sounds interesting and promising. I guess Shirley or Sarah, if there's anything you'd like to pose to Dr. Parsons, we can also take the time to do this now as well.
I think the presentation was very interesting. I think that you did a phenomenal job, as Caitlin said, in terms of explaining things, which is why I guess we haven't gotten a huge number of questions. And I think that the way you presented the information in terms of the sponge and various different models was very interesting. We do have a question. When you talk about treatments early on, does that mean before symptoms?
Yeah, it's a great question. In an ideal world, yes. But even when symptoms start, I think just in general, the earlier the better. But even when symptoms begin, the right treatment can certainly halt things, halt progression in its tracks, and also have some reversal. So the cell loss, a bit of cell loss can be tolerated, but I guess the main point is that we don't want cell loss to get so far advanced that it becomes near impossible to replace those cells. So in an ideal world, yes, as when you're a teenager, you'd have there be some excellent test that would say, yes, without a doubt, you are going to get Alzheimer's disease. Then you start treatment as early as possible. That would be the ideal world. I think the earlier detection is better, but I certainly think that it's never too late for some drugs to have beneficial effects. Absolutely not.
Excellent. Any other questions from the attendees? Okay. Well, I'd certainly like to thank you for your time in terms of presenting this information to the group. And there were over 24 plus attendees today. So I think that you certainly enlightened a lot of people in terms of the research that is being done. And as the Alzheimer Society, I think any research that gets us closer to the stages of a cure is beneficial to the organization. So I know I'm from Newfoundland, but I'd also like to thank you from our province and from across Canada for the work that you're doing for people with dementia. And hopefully this will be one piece of the pie that gets us closer to the cure someday.
Great. Well, thank you very much. And thanks for everybody for attending and the comments here. And thanks again, Sarah and Caitlin, for organizing.
Thank you so much, Dr. Parsons. I can see now that there's a few comments coming through just from attendees saying how excellent the presentation was and that it was very easy to understand. And as Shirley mentioned, it's such an enlightening thing to learn about, and any type of information that researchers with your expertise can provide is very valued by the society. And I know from those who utilize our services that this is something that will mean a lot to them and the effort that was put into such study. So I will just move on, Sarah, to the final slide, please. Yeah, and just another comment saying that this presentation really did help individuals understand a very complex issue. And again, just wonderful job breaking that down for everybody.
Great. Thank you, everybody. Appreciate it.
So I'd just like to mention that we do have another upcoming ASRP Exchange presentation in September. This will take place September 29th and will be led by ASRP funded biomedical grant recipient Dr. Michael Jackson from Manitoba. This presentation is titled Identifying Prospective Therapeutic Targets in Alzheimer's Disease, Understanding the Cascading Effects of Cellular Deterioration. And this, as mentioned, is on September 29th and will take place at 12 p.m. Eastern Standard Time. The link is provided at the bottom of the slide and is also available on the Alzheimer Society of Canada website, ASRP Exchange webpage. I will conclude today's webinar here. Thank you once again to everyone who attended. Thank you, Shirley. Thank you, Dr. Parsons, and thank you, Sarah. And I hope everyone enjoys the rest of their day.
Hey, you too. Thanks a lot, everybody. Bye.
Thank you.